you. So you said you're starting your CNA course in like 2 weeks or something?
Yeah. Next Monday.
Okay.
The Monday following. Ending.
Okay, gotcha. What? Yeah. Blessing, are we still, thank you, are we still meeting to this?
After this meeting.
Okay.
I mean, after this class, yes, sir.
Good, good.
Yes, sir.
Nice certificate there, Brady. You gotta frame it. Hey, how come you're not wearing your ribbon?
Yeah, here.
Okay.
I'll probably put it somewhere. We'll see what happens.
So what are your plans for the summer?
Work, go on some trips, and that's about it.
Alright.
So, excited though, going to North Carolina.
Oh, that's nice, that's nice.
It's all of our roommates and our girlfriend and stuff like that. So we went freshman year, but then when you go last year, it was like dad or kids.
Oh, okay.
It'll be fun this year though.
Good, good. Are you a junior or what? Okay. Then what are you looking at for after graduation?
I want to do something with obviously environmental science or like animals, so biology, so like conservation and zoos and other stuff like that. So, I mean, I'm gonna be working with the DNR this summer.
Okay,
but it's
good,
not for,
good.
It's just, it's just like around the parks and stuff like that. But it's a step into
Right, right.
connections. So.
Yeah, absolutely. That's
Yeah.
what, you gotta start somewhere.
Yeah.
Look good. And how are you two doing today? Good, good. I'm happy it's Friday. I'm happy it's Spencer's lined it up here. So you're gonna continue working at like a vet clinic this summer?
Yeah, I am. I haven't got any email back from the one yet, but I have like 2 other options.
Okay, okay.
Just around the area here. And we're getting puppies soon.
Oh,
So,
that's sweet.
That's taking up a lot of my time this summer.
Right. Where are you currently working at a vet clinic?
I was last summer. The one that hadn't applied to me yet, but
Right, okay. So hopefully, same place or different?
yeah, same place.
I know, this is awesome.
Fieldstone Veterinary, it's over in
Alright, well, that's good that you got the inside around. Ryan, did I already ask you what you were doing for the summer?
No, I'm gonna be working at a tackle shop over in Port Clinton, off of Lake Erie.
Yeah, that's awesome. I mean, I imagine they get a lot of some serious fishermen over there.
There's a big tournament that's gonna be coming through during the summer, so they'll stock in a bunch of walleye guys, bass guys.
Alright, very good. Where's your pen?
In my bag. There are both of them in here, also connected to the certificate itself.
Oh, okay, alright. Well, super cool that you stepped up to be an officer, so congratulations with that.
Thank you.
Do you have plans for the summer? Are you working or?
Working, visiting Florida, family.
Right, yeah.
We're going to a concert.
All right. Sounds good.
What state park or?
How are you today?
Good, how are you?
Good. Any plans for the summer?
I'm going to California and Hawaii at the start of May, and then I'm going to Wisconsin for a month, so I'm all the time. No, in Wisconsin, my parents live in California right now, so I'm gonna go live with my boyfriend's family in Wisconsin. And then I've been doing this camp since I was like 7, so this is like how I've grown up doing it, now I'm so a little closer for more in my life.
So I applied.
I'll be doing that.
Work with.
Oh, that's great.
Yeah.
For fishery biology.
Oh, we're good.
Yeah, no, definitely, my keep me active, I get a nice tan, so you gotta go outside, you gotta be alright, you gotta keep kids entertained for 7 hours. Yeah, keep it going.
And I'm just like, in the mornings, I've just been like, not talking because I'm all sluggy, I'm like, oh yeah, I don't know if you were in bed this morning, I was trying so hard not to like just talk, sounding wrong ever, and I felt so bad for the first one. And I was like, no, like, I'm not sick, I promise, I'm not.
Good, I was good. I'm working on that steak. So I was working on it right there, but I left to be at home, so. Getting things together for patient contacts and whatnot.
Yeah.
Should like $10,000. That'll be alright. But we only contact like 5, so I wonder if somebody
I'm just gonna pay attention.
no avoids. I think they only contact like 5. Some places, so I've read.
Boyfriend works over at
Up to 5,000 hours they'll count for, so, I don't know, maybe they'll just give me extra credit, I don't know, right?
Are you from Michigan?
No. You can do the hand thing, I do know that.
Right, I went to a job interview this morning at a restaurant down in just.
Oh, is it here in Florida?
Yeah.
But I
Okay.
think you really want me to get.
Oh, yeah, okay. Nice, that should
I
be
should
cute.
focus on that this summer, is work on an ambulance or volunteer in Madison Township or something. Schools seems to like me, the chief there. So,
Hope you enjoy it. State Park works for me.
that'd be cool,
How promising.
right?
Oh,
There's Liz.
this from mom.
But it's like, like, it was starting this one. But they didn't have any issues. Am I supposed to do
That's like a
Feeling I go.
I wrote mine down.
Oh, we got
No, shut up. No, not a single day. Yeah, so you're just kind of sitting there for 2 hours, but.
Oh, sorry.
Oh, it's okay. We were making ourselves entertained because it was still like slime and we were doing the experiments for each other. But
Okay. Well,
yeah.
maybe next year I can help you.
2:30 till.
We said it to like 8 other
Because it's like,
That's.
some of these schools, they have like STEM night that you go to.
I just like, I
They'll have kids there and all that, all the discovery and stuff like that. So maybe that's something that.
Yeah, maybe we'll do a reverse next
Yeah,
time.
reverse next time, yeah.
One thing that they would randomly make. Sure, that's nothing.
Well, good afternoon, everyone. I appreciate that you came to the last, the last lecture for the course. I only have 3 slides to share with you today, so hopefully we should be able to end a little early. That's more time for studying, right? So that'll be a good thing. So, so here's one of the 3 slides. So remember that we're talking about how these, how we interact with these microorganisms. Some of these organisms cause disease, and they cause damage by producing toxins. So this first slide is going to give us information about exotoxins. The next slide after this one is about endotoxins. So there's two classes of toxins that these microbes can produce. Exo and endo. So I know they sound very similar, but they're actually very different. So this is about the exotoxins. So exotoxins are named because these are produced and released outside of the cell. So these are secreted by both gram-positive and gram-negative bacteria. And then the idea is that those toxins, once released from the cells, bacterial cells, can then damage our cells. Okay, so a couple of points to note. One is that these toxins are very dangerous for our cells. Hence, you'll see on the slide that we have the highlight that it's highly toxic. Fortunately, these toxins are made out of proteins, and proteins they readily unfold when exposed to high heat. Anyone remember that there's a term for when these proteins unfold? It is denaturation, that's right, yep. So because these toxins are made out of proteins, we can destroy them, make them inactive using high heat. So that's the reason why we like to boil things. We can use an open flame to sterilize. So by doing those actions, we can protect ourselves from these toxins if they are present. Now that doesn't mean that we can once in our bodies that we can jump in a vat of boiling water, but the idea is that it would destroy the toxin, make them inactive before the toxin enters into our bodies. Okay, well, what happens if the toxin, well, how else can we protect ourselves? Well, it's through immunization, through vaccination. We have a term here called toxoid. So we can actually create, versions of these protein toxins that are not active, they're not functional, they're not harmful to our cells. And then we can use that as a basis for inducing those memory cells in our bodies. So we can use them as vaccines. We can inject ourselves with these modified versions of the inactive form of the toxins, what we call toxoids, introduce that into our bodies so that our cells will generate the antibodies to it, so that if we are exposed to the genuine toxin, then our bodies will be able to react immediately to it and inactivate it. So our antibodies can coat it, and so that the toxins will not come in contact with our cells. Okay, and then the last item here is, well, we can also, if exposed to the toxin, we can be injected with a solution of antibodies that have already been created to bind to the toxin and will protect ourselves from it. So it's those antibodies that are collectively form what we call an antitoxin. So it's antibodies directed against the toxin. So that will protect us too. So there's a number of measures that we can, because we understand, the chemical nature of this kind of exotoxin, a number of different ways we can protect ourselves. Number one, using proper, I guess it's like sterilization methods, like using excessive heat to deactivate and destroy the toxin before it enters our bodies. You know, immunization. You probably heard of this one here, diseases called tetanus and diphtheria, and there's like a combination vaccine that has been developed. Anyone remember the name of that vaccine that you get when you're young to protect you from tetanus and diphtheria? Yeah.
I don't know, I think I'm about to lie here, but I'm thinking of BCG.
Okay, we'll go back here. Tdap, that's right. I thought someone had that one on there when they did that bacterial presentation. Okay, so, all right, very good. So, you know, it's a childhood vaccine that, you know, you get in order to protect yourself from that toxin. And then even if all that fails, you can be treated with an antitoxin. So those are all good things. All right, next we're going to learn about endotoxins. So I'm going to skip this slide here. So here's information about endotoxins. And they're named endo because these are actually, the toxins are actually components of the bacterial cell wall of gram-negative cells. So I don't know if you remember, but gram-negative bacteria have like an outer cell wall that includes a lipid layer. And within that lipid layer, there's a lipid molecule called lipid A that's a component of the lipopolysaccharide. So it's actually the lipid that's harmful to our bodies here. So the idea is that if we were to kill these cells, you know, the cell walls would break apart, the membranes would fragment, and then the lipopolysaccharide and that deadly lipid A would be released, and then could, you know, potentially come in contact with our bodies and cause damage that way. These toxins are less toxic, less harmful to our cells than the exotoxins are. I think we already learned before that, you know, that a systemic inflammatory response can be it can be deadly, it can be lethal. So what do we mean by systemic? Widespread, that's right. So if the toxin moves, you know, away from, you know, potentially the initial location of where it was produced and just spreads through your blood system to various parts of your body, you could get inflammation occurring in multiple places at about the same time, leading to a, you know, rapid loss in blood pressure, creating this condition referred to as septic shock. Now, because it's made out of lipid and not protein, that means that this toxin is not destroyed by high heat. So it's very heat stable. So because of that, because of the chemical properties are different from those of the exotoxins, you know, the approaches about how to protect ourselves are going to be different, because we just can't boil things anymore, because boiling will kill the bacteria and result in the release of these exotoxins, and those toxins will not denature like the proteins ones do. So instead, the approach is to, you know, prevent growth from the beginning. So trying not to have any bacteria present. So we're talking about, trying to create sterile conditions. And then there needs to be monitoring. There needs to be some testing procedures in place in order to determine whether there's any bacteria present or not that may be, potential sources of the endotoxins. So that brings us to this assay, this test. Interestingly, when I grew up, in my household, there was this shell of a horseshoe crab. You probably have never seen a horseshoe crab before, but the one that, they can grow pretty big, but I guess it's, I don't even know how to describe this thing, but trying to imagine it's like the main body of like a lobster, but with an extended tail coming off of it that's kind of like a possum. It's kind of weird looking. But anyways, that horseshoe crab is limulus. And what scientists discovered is that there are some cells in the blood of the horseshoe crab. Those cells are called amoebocytes. And if you break open the cells, that solution is the lysate. And the horseshoe crab has a defense mechanism against these endotoxins, so that when they encounter the endotoxins, the lysate will actually create this gel-like solution to kind of bind up and prevent the toxin from working. So scientists kind of stumbled upon that observation and actually developed a test, which is what we call an assay. It's a chemical test to determine whether the endotoxin is present or not. So the idea is that if the endotoxin is present, you expose it to this lysate, it'll create this gel-like reaction, and that'll tell you, okay, that substance has toxin, and let's not use it. It could be a food product, it could be a medicine or something like that that would potentially cause harm to you if you were to ingest it. Okay, so why was this horseshoe crab sitting around my living room? It was literally in the living room. Well, it's because my dad was a microbiologist, and that was his training, and he worked in the pharmaceutical industry. And for about 10 years, he worked on this limulus amoebocyte lysate assay. So that was the little history lesson there. All right, any questions about these two types of toxins? Yeah, exotoxins, endotoxins. Okay, last slide coming up here. And that is kind of related to the pharmaceutical industry, because that's one of the items listed here. Now, whenever I teach, I always like to try and make connections between the information that's in the textbook and try and relate it to some practical uses and try and relate it to your own life. You may have noticed when you gave your presentations about your bacteria that there were a whole bunch of questions there about, well, are they related to any diseases? Do they make any products that are useful? Do they play a role in our environment? Stuff like that. So this is how we're going to end the course, is to talk about, how can you use the knowledge that you gained in this course as potentially a way to help society and get paid for it. So that's what we mean by a career, but a career with purpose, right? Because life isn't just about making money, it's about service to others. It could be to help your family, your friends, your community, your country. Okay, so there's different ways that you can do that. So some of you may, probably already are leaning in the direction of working in the medical field. And with your microbiology knowledge, you can actually work in hospitals and in various kind of laboratories, doing tests. And these are tests for things like those toxins, for example, whether they're present or absent or not. It could be tests for diseases, like doing blood tests to determine, if it's a bacterial or viral infection, what kind of bacteria and virus are they? Those are really important to know that kind of information in order to apply appropriate treatments for diseases. All right, and I mentioned that my dad worked as a microbiologist for the pharmaceutical industry. So these are just some examples of some of the different companies that exist. Have you ever, any of you are like Breaking Bad fans? Okay, yeah, it's okay. It was like I think it was like a Netflix series, and supposedly this high school chemist, Walter White, needed money to pay for his cancer treatment, so he ended up, synthesizing, I think, methamphetamine. What was that? Like some kind of meth, methamphetamines or something. And he got all caught up working for the cartel. But anyways, in that series, you guys remember seeing his underground lab, where he had these huge stainless steel vats, fermentation tanks, and all that. He was working with. Okay, so trying to imagine that. Because that's actually very similar to what you would actually see working in the pharmaceutical industry. Those are fermentation tanks growing microorganisms, like those that produce antibiotics, those that can produce useful products that we can use as medicines. But instead of Walter White's underground lab, these things are legal. It's lawful to do this, and it's above ground. But things are very clean. You don't have to wear a respirator all the time either. So some of you may be interested, in the development of new medicines, vaccines like the COVID-19. Vaccine that Pfizer, created right within the state of Michigan here. Okay, my dad did not start off working for the pharmaceutical industry. He actually started off working in the food industry. So there's a number of companies here that actually employ microbes to make food. Can you think of what some examples of foods that are produced with the aid of microorganisms? Yogurt. What else? What? Kimchi. That's right. What else? Fermented beverages, that's right. I'm surprised you didn't catch that one. But yes, beer, wine, yes. Dairy, think cheese. There's a bunch of those kind of different food items that involve microorganisms. So that's a huge area as well. And then Brady, you're interested in the environment, right? So there's actually jobs, with the government, like the Environmental Protection Agency, the DNR, There's also, companies that have to monitor both water and air to ensure that, that they have safe limits of microorganisms present. So we have monitoring of our public beaches for contaminants, monitoring of our drinking water, monitoring of the air that we breathe. I think we kind of take those things for granted, that the water is safe, that the air is safe, but once you start traveling around the world and start visiting some of these third world countries, no, they don't have that kind of monitoring or controls in place. And the last one is, working with the government. It could be state, it could be local government or federal government. The Center for Disease Control is our national federal organization that is involved in pandemic, epidemic kind of outbreaks and monitoring those kind of things. So, one of my former students actually works for the state of Michigan, doing DNA testing for different kinds of microorganisms related to food outbreaks. She does COVID-19 wastewater monitoring. She also uses her talents to look at crime scene evidence that's left behind. So there's a lot of different things that you can do with your knowledge here. So you probably heard of the expression, knowledge is power. Have you ever heard that one before? Okay, well you heard today. Knowledge is power. So use your power for good, right? Not evil, but use your knowledge for good. To feed yourself, take care of your families, help others, serve your country. All right, that's how we're gonna end the course here. So, any questions? All right. Lastly, when are we meeting next? Monday. Yay, 1 o'clock. In this room for our last exam, over the last two chapters 15 and 16. Don't forget to upload your notes to Blackboard there. Yep. So I will see everyone on Monday.